I'm John Epstein, the Executive Vice Dean and Chief Scientific Officer at the Perelman School of Medicine, and it's my great pleasure to welcome you to this discussion and to welcome you to this virtual homecoming. This panel will be entitled Penn's ImmunoRevolution, Rising Stars and Next Generation Therapies. You'll hear about some of the exciting work, part of the ImmunoRevolution at Penn, where we're really at the forefront of translating basic fundamental discoveries at the laboratory bench to be new therapies that affect patients' health. The ImmunoRevolution, I think, began at Penn about 20 years ago with critical gifts from Barbara and Edward Netter and Leonard and Madeline Abramson. Those gifts allowed us to recruit Carl June and others who have transformed the care of cancer patients by discovering how to harness the power of the immune system to cure pediatric leukemia, work that's being extended now to lymphoma and to other types of cancer. And in fact, one of the most exciting things that I see happening now is the extension of these discoveries to the treatment of diseases beyond oncology. We may be able to treat Alzheimer's disease with this sort of approach. And in my own laboratory, we're working to treat cardiac disease with engineered T cells. The ImmunoRevolution is really affecting all aspects of medicine, and Penn is leading the way. Penn has one of the largest immunology communities of any academic institution in the world, and they're working to apply our new knowledge to the development of new vaccines, perhaps vaccines to prevent cancer in the first place, the development of small molecules that can modulate the function of the immune system, and of course the use of monoclonal antibodies to treat various disorders. One of the great strengths and hallmarks of Penn has been and continues to be the ease with which we can collaborate across institutions and to find discoveries at the interface of disciplines. This is particularly true among scientists working together between the schools of engineering, medicine, the vet school, arts and sciences, the nursing school, Wharton, etc. And our physical proximity makes that easy as well. One of our lead focuses for future research here at Penn in the next 5 to 10 years is, in fact, in the area of immune health, extending the ImmunoRevolution so that we learn in greater and greater depths how to read the language of the immune system, how to use it to predict response to disease, and how to use it to direct us as to which therapy may be most beneficial for any given patient. And I couldn't be more proud of the advances that this research community are making. We just had our 12th FDA approval of a new agent for cancer therapeutics and diagnostics, in this case, a new PET imaging probe to be used in patients with metastatic breast cancer. Going forward, I think what we'll see is a greater understanding of the power of both harnessing the immune system to treat our own diseases, how our own body can help to keep us healthy, and how we can use the immune system like a sensor of illness, reading out and telling us even before we have symptoms whether we might be getting sick. And I think you'll enjoy listening to the upcoming discussion. So welcome. Thank you for joining us, if remotely.
Well, thank you, John, and welcome to all of you for joining Penn Medicine Homecoming. Welcome home. You're tuned in from all over the country, in fact, all over the world. My name is Bob Vonderheide. I'm the director of the Abramson Cancer Center here at the Perelman School of Medicine and the University of Pennsylvania. And it's my privilege to moderate today's panel. We're going to talk about the ImmunoRevolution, as John mentioned. And here in this city of Philadelphia, we know a revolution when we see one. And we have a power-packed hours program for you today. And like every good revolution, we're going to have a fireworks show. And why is that? Because we're going to talk about the power and the promise of using the immune system to not only address disease, but to prevent disease. Here at Penn Medicine, we are leaders in this area, as you know, working very hard on novel therapeutics, the incredible revolution from Carl June and CAR T cells we all know about, but we're also working on understanding the immune system, as John said, in a project we call Immune Health, where we understand every patient's immune system like a fingerprint that we can address. So the structure for today will be to fulfill my childhood dream one day to be a TV talk show host. I'm going to be joined by 7 wonderful scientists, all Penn faculty members. They come from all walks of life. We're going to go far beyond cancer. We're going to meet senior scientists. We're going to meet scientists who are just beginning their research programs. Their biographies are available to you in the materials when you signed up for this, and I won't go through the many, many accolades and all the accomplishments of our esteemed panel. I will tell you who they are. These faculty members are Dr. Amit Bar-Or, Dr. Caitlin Byrne, Dr. Joseph Frietta, Dr. Sar Gill, Dr. Shannon Maude, Dr. Ali Naji, and Dr. Drew Weissman. And what I'm going to do is go around and introduce them to all of you and chat with them for a bit. And then we're going to turn the bulk of today's session to your questions, which you can place in the chat room. Many of you have placed them already ahead of time in the portal there. So without further ado, let's go straight to a topic literally ripped from the headlines 2 days ago. And we'll say hello to Dr. Drew Weissman. Drew is professor of infectious disease, has been at Penn for some time, where he runs a research laboratory. Now, on Monday, as all of us know, Pfizer Corporation and its collaborative biotech company announced very promising results with a prevention vaccine to address COVID-19. And what's not so well known, but we're here to talk about today, is that vaccine, and we do hope it makes it across the finish line, that vaccine is based on a technology using RNA, not DNA, but RNA, in a certain way that Dr. Weissman discovered here in his Penn laboratory many years ago. It is his technology that has been licensed for the further development of this vaccine. So, Drew, welcome. We're so happy to join you. I know you've been on the phone and the radio and the email all day today with all the major news outlets. So thank you for sharing time with our Penn alumni today here at Homecoming. Let's just cut to the chase. Drew, you're a world expert on this. You've been working on coronavirus 24/7 since it became a problem a few months ago. Your laboratory studies were promising that this vaccine would work. Just from your point of view, is this what we've been waiting for? Is this the news we were hoping for?
So I think this is fantastic news. As a vaccinologist, I can't remember ever seeing a respiratory virus vaccine that had 90% efficacy. We're usually thinking about flu that's 40 to 60%, and we're happy with that. This is unbelievable. That's just incredible efficacy, and I think it's probably going to be a wonderful vaccine.
Well, we're all hoping and wishing it the best. Drew, every great breakthrough has its moment when it was created. I want you to take us back to your laboratory around 2005. You were working on the Rna molecule and how it's and how dendritic cells, a certain immune cell, responds to it. Can you tell us back then what you discovered that was eventually licensed that has now 15 years later turned into a vaccine for coronavirus when we never even imagined it?
Sure. So Katie Carico and I had been studying the immunology of RNA for many years. And so RNA has always been thought of as a great way to deliver proteins. But the problem with it is that it's so inflammatory that we often killed the mice, which doesn't work well for a therapeutic. So we studied the immunology, and what we found is that by changing one of the bases in the RNA, one of the components of the RNA, with slight modifications, we could get rid of that immune activation inflammatory potential. And what was also surprising is that when we did that, we increased the amount of protein that RNA produced by upwards of 1,000-fold. So not only did we make the RNA safe, we made it 1,000 times more potent.
It's amazing. My understanding is RNA is ordinarily so inflammatory that you get this immune response that's not productive. But by altering it, you're able to then use its properties to actually start an immune response, in this case against coronavirus. That's incredibly clever. It means perhaps, Drew, that we can use this for a vaccine for many other indications as beyond coronavirus. Isn't that right?
Yeah, so we've got clinical trials already set up and ready to start for genital herpes, influenza, HIV, norovirus, C. diff, and malaria, and others on the way. So even before coronavirus, we were ready to go with a bunch of different vaccines.
Yeah. And the material we're talking about here, RNA, it's different than conventional vaccine. It would be the first time to use RNA in a commercially available vaccine. Is RNA hard to make? Is it hard to manufacture? Is it expensive?
Well, that's probably what attracts pharmaceuticals the most. Making vaccines is very labor intensive, very expensive. Making RNA, it's a two-step reaction. It's simple. It's one tube to make the RNA, one tube to put it in a lipid particle, and you're done. The cost of RNA vaccines is much less than other vaccines.
Yeah, that's and I guess that explains why we were able to go from not even knowing the virus in January to mature clinical trials here being reported in November. Really unprecedented speed. It reminds me of sometimes we say, these breakthroughs are 20-year overnight sensations. So you say it's easy, you make it sound easy, and to you it is easy. And we don't want to jinx things. I think we have to keep our head down. What do you think the next step from here is? What can we expect next, Drew, in the news or scientifically?
So I think we're now convinced that the Pfizer vaccine works. The next step is the Moderna vaccine. After that, to me, as a clinician, the most important next step is to get it out to the people and to have people take the vaccine, because it isn't going to do much good if people don't want to take it.
Yeah. And are you concerned about safety of this formulation?
No, I think the safety, so the vaccine gives local side effects. People's arms hurt. But other than that, there have been no bad side effects. I think it's a completely safe vaccine.
Let's hope so. And I think I share your confidence. RNA is not toxic and has never been shown to be toxic. So another reason to take advantage of it. You mentioned another company, Moderna, and just to point out, Moderna also licensed the Penn patent that is at the heart of their vaccine as well. So, Drew, thanks for joining us today. Let's that's certainly an immune revolution, and we look forward to hearing more, and that this vaccine is one of the answers we've all been waiting for. I'm going to shift attention to some of our other panelists as well and introduce the other 6 scientists who are joining. Next is Dr. Shannon Maude. Dr. Maude is an assistant professor of pediatrics. She's an attending physician at Children's Hospital Philadelphia and has been involved in CAR T-cells therapeutics for acute leukemia, patients with acute leukemia for many years. She is now the international leader of one of the largest trials in the world to further advance this therapy, building on the work Carl June and Steve Grupp had done previously. So Shannon, thanks for joining. Can you can you just give us some insight into the impact that this is having in caring for your patients?
Thanks for having me. I think the impact is nothing short of amazing, really. This is giving children their childhood back. I can remember in my training being in situations where children with acute lymphoblastic leukemia had relapsed multiple times or had relapsed and not responded to the chemotherapy or transplant or multiple intensive therapies had failed them. And we really did not have effective options. We had very limited options. And now children in that situation really do have an option that could potentially, dare I say, cure them of their disease. And so I think what is so wonderful about this is not only are we seeing remissions after chemotherapy has failed, but we're seeing remissions lasting years. And we're seeing children come back to us and having their parents say, I haven't seen my child act like this since before they were diagnosed.
Can you imagine? I don't know how. Those must be some very emotional meetings. Right now, as you said, this has been approved for children who've been through a lot of other therapies, sometimes even a bone marrow transplant. So if we keep working hard at this, could we advance the CAR T-cell technology to a point where we don't need bone marrow transplants for children with this disease? I'm just really going out there.
I think that's what's very exciting, that I think that is possible one day. And we are doing a trial right now to hopefully answer that question, where we are incorporating this therapy early on into therapy for patients who are newly diagnosed and unfortunately have not had the response that we would like to see to initial chemotherapy. And so they are at very high risk of relapse. And so we're incorporating CAR T cells after just 5 or 6 months of chemotherapy and may be able to use this as the definitive therapy and not need to go to bone marrow transplant. So I think that's a question that we may have the answer to in the coming years.
And the final thing I'd ask you before we bring in another guest is, right now we have one FDA-approved product, but do you see that this is just the beginning? I know you're working on many novel formulations as well. You have confidence that we'll, in 3 years' time, we'll have something even better?
I certainly hope so. And I think there are a lot of therapies in the pipeline and currently in clinical trials that are very promising. And what I hope to work on, and I think we're most excited about, is how can we improve even further upon this and have those rates of durable remissions be much higher.
Well, we're with you on that. It's important to point out to our audience that CAR T cells are they're made from one patient at a time. And there's not just one type of CAR T cell. There's some that might work in leukemia and some that might work in other diseases. And I want to bring in our next guest, who is Dr. Sar Gill, and we'll move the video around. You'll see him. Sar is Assistant Professor of Hematology Oncology based at the Hospital University of Pennsylvania. Welcome, Sar. Thanks for joining. And see what I mean? It's like a talk show, okay? And Sar is also working on CAR T cells, but for adult patients, and was involved in some of the early studies as well. Sar, following up on what Shannon and I were just talking about, how are we going to, and what are you doing to advance this technology, CAR T cell genetic engineering cell therapy, to other diseases and other situations?
Thanks, Bob. Pleasure to be here. So the way I see it in terms of the message that I would want to share and convey to people in the audience is that having established that this platform works, and by platform I mean technology, the technology that Shannon was talking about just recently, what that allows us to do, and what I've been particularly interested in doing over the last few years in my lab at Penn, is to expand it beyond ALL, to expand beyond B-cell malignancies, to expand it beyond cancer potentially, and I think you heard a little bit about that from Dr. Epstein before. The two ways to sort of think about this are just as the sort of discovery of steam engine led to trains and ultimately to cars and planes and speedboats and all the things that those have led to and allow us to do, finding out that the CAR T-cell technology can cure perhaps some people of their leukemias now has exploded out into the possibility of us treating other cancers and maybe other diseases as well. Perhaps another way of looking at it, this exact thing, is having shown that the smallpox vaccine back in the 19th century works now has led, albeit 200 years later, to what we talked about with respect to an mRNA vaccine for COVID. That's what I mean by showing that the technology works and then expanding upon it. And so specifically with respect to your question, Bob, maybe two small examples. One is we have developed a technology to put CARs on macrophages, which are another sentinel immune cell in the immune system, and have actually spun this technology out of Penn to a biotech company that was founded out of Penn, and using that technology to try and treat metastatic solid tumors, which are a big problem, obviously, and one that has to date not really been served well by CAR T cells.
Solid tumors, you mean like what? Solid, what do you mean by solid tumor?
So thank you. So solid tumors such as lung cancer, breast cancer, ovarian cancer, those sort of malignancies that have proved incredibly difficult to treat, and pancreatic cancer that you'll hear about a little bit later. And the second sort of facet of that, having developed macrophages, CAR macrophages, genetically engineered macrophages for the treatment of cancer, and put it into a company that is now about to open a clinical trial at Penn in the next month or so. Having developed that technology, it then led some people in my lab to pause for a second and say, well, if we are able to redirect macrophages to do what we want them to do, what else are these cells capable of? Well, unlike T cells that are very good at killing, macrophages can also engulf particles. They can engulf misfolded proteins, which actually paves the way potentially to treating some other really difficult diseases that are driven by misfolded proteins. And what I'm talking about are some neurodegenerative disorders such as Parkinson's or Alzheimer's disease. And that's another part of what my lab is working on at the moment.
Well, we'll get some thoughts from Dr. Bar-Or about that in a few moments. It's interesting, Sar, we talk about cell and gene therapy. So then you have a couple of choices. You can change how you do the gene therapy, which we talked about with Shannon and you mentioned, but we can also change the cell. So we've been using T cells, and you're saying there's another cell, macrophages, that are also cancer killing and disease protecting that we can also use. It's clever. You're clever. We're going to come back in the question and answer for more conversation with you, Sar. Thanks very much. We're going to introduce Joe Frietta. Joe is a new member of the faculty. He's an assistant professor of microbiology and has been at Penn, did some of his work with Carl June early on, now has his own independent laboratory. Joe, what I wanted to ask you and help the audience understand is that we don't just do clinical trials and hope for the best. We then get samples back from these patients. They partner with us, provide samples and biopsies, and we study them to find out what went right and what went wrong. And sometimes we find patients who did extraordinarily well, and there's secrets there. And some patients who did not well, and we want to know why things didn't work. How can we make it better? Can you, I know your work is focused on that. What can we learn from patients once they go on a clinical trial?
Sure, thanks, Bob. That's an excellent question. And so even though I'm a basic scientist, I really focus on bench to bedside research, and then, as you mentioned, rapidly translating our discoveries in the lab to state-of-the-art clinical trials that bring the newest treatments to our patients here at Penn, and hopefully beyond Penn. And then also taking what we learn from our patients back to our labs to find new ways to fight cancer. So as you said, why does the therapy work? Why does it not work in some cases? And so I think a good example of learning from our patients involves some of the retrospective studies that we do. So more specifically with CAR T-cell therapy of advanced leukemia that we heard a little bit about from Dr. Maude. In my case, I'll talk about chronic lymphocytic leukemia, but we found that patients with stunning responses to the treatment, they turned out to have an existing ample supply of memory T cells. So we're coming back to T cells here. And these cells prime the immune system to watch for rogues, right? Like cancer cells. And although there are several types of memory T cells that have been identified, we discovered a completely new subset by doing those important studies, by analyzing the results of 41 patients who received CAR T cell therapy. And these are CLL patients again. So, and beyond that, we found distinct protein markers. So these are sort of a fingerprint, if you will, on this particular subset of memory T cells. And then we validated the fingerprint by using it to forecast responses in additional leukemia patients who are not a part of the initial analysis that we did before these patients underwent CAR T cell therapy. And what was pretty striking is that, all of the patients who experienced complete and very deep remissions were correctly predicted using our new immunological biomarker. So I think, this is a perfect example of learning from our patients and potentially using that information to improve outcome.
This would enable precision CAR T cell therapy, where you don't just guess, I think this might work in this patient. You say, no, you need flavor A, but you need flavor B. And I want to pick up on a phrase you used because it's really important. You said memory T cell. So we think about memory being part of the nervous system, but you're talking about these immune cells having memory. Can you, why is that important?
Yeah, so CAR T cells are, and you alluded to this earlier, Bob, they're a living replicating drug. So unlike targeted agents, unlike drug compounds, they can go anywhere, do anything, and most importantly, make decisions and sometimes remember what they've seen. So even after you have a primary response elicited or driven by these CAR T cells, a fraction of them sort of remain behind as sentinels. And again, they remember these rogue cells that they've seen before, and if they encounter them again, they can kick into action and sort of take care of that.
Well, I would add, chemotherapy cannot remember.
Right.
Radiation therapy cannot remember, but CAR T cells can remember. You get this living drug, which is your own cells re-engineered, and if it can remember, if the immune system can remember what cancer looks like, if the immune system can remember what coronavirus looks like, then it can do that job while we go about our regular life and stay healthy. Does that make sense?
Absolutely.
Right?
Absolutely.
Well, it's really impressive, Joe, the way you do rigorous science based on patient samples. It's a really difficult experimental model, and I know meeting patients how much they wish to partner and hope that we can learn the most from when we treat them. We have incredible patients here, and you've benefited from their generosity. So Joe will come back. I'm going to introduce our next guest, who is Dr. Ali Naji. Dr. Naji is the J. William White Professor of Surgical Research. Ali's been at Penn for some time and has been a pioneer in transplantation. But what we want to talk about today is not transplanting an organ like a heart. We're moving away from cancer, but a cell that has something to do with diabetes. So, Ali, tell us what you mean by islet cell transplantation and how could it potentially be a cure for type 1 diabetes? Welcome.
Thank you, Bob, for the opportunity. And it is a privilege to be in my Penn family, the current one, and also the alumni. Bob, each one of us are made of trillions of cells. Each cell needs energy, metabolism, and glucose is the best premium gas to provide energy for the cell to have this enormous cooperation to give us life. The glucose is the best molecule that the cells rely on. What is important, the entry of glucose to be utilized by each cell is tightly regulated by insulin. Insulin is at the gate. It will allow you whether the glucose is going to get inside the cell or preclude it from getting into the cell. Now, unfortunately, in diabetes, the cells that produce insulin, which is called beta cells, in the islets in the pancreas, are selectively destroyed by the immune cell. There's a lot of wonderful research is going on. What triggers that? Somehow the immune system has gone through a dysregulation to do this. It's analogous to a friendly fire that unfortunately in a defense also leads to mortality of the friendly participants. Be that as it may, when the beta cell or islets, which we only have about 1.5 to 2 million, are destroyed, there is no insulin, so glucose cannot get into the cell. In reality, our cells, doesn't matter if it's a brain cell or the heart cell or the skin, they suffer in the midst of plenty. So here's a lot of good nutrition, but it cannot get into the cell because of the lack of glucose, lack of insulin. So affected individuals, they need daily injections of insulin to provide some degree of entry of this glucose. But unfortunately, it is not as precise and it's not as accurate as the amount of the insulin that's produced by the beta cell. Beta cell is very smart, knows exactly how to produce. So the concept is next spring would be the 100th anniversary of discovery of insulin in the University of Toronto. And despite the fact that the insulin was a miracle, however, it really saved human beings from mortalities, but left diabetics with a long-term complication, number one cause of blindness, heart attack, and a host of kidney failure, etc. So the concept was conceived at Penn. If the insulin injection doesn't work, why not just take islets from the pancreas of a donor and transplant it into a diabetic recipient? For the past 30 years, I've had the privilege of going through this journey, providing its advance from the bench to the bed, to the fact that we have the best result of islet transplantation in human under the auspices of NIH. Let me just share with you that this journey would not have been possible without what Dr. Epstein and Rob mentioned, this permissiveness, this spirit of collaboration in the Penn family. And more importantly, the breakdown of the silos or barriers from which department we come from. The Institute of Diabetes, Obesity, and Metabolism, really the mission is to bring in talents, focus on this complex disease with devastating long-term complications. So I believe the phase 3 trials has proved the efficacy of this. And our goal at Penn is to use this to become a standard of care.
Wow.
And one other point that I would like to mention is how permissive and collaborative Penn is. We have now utilized the ImmunoRevolution led by Carl June and a number of his associates to really make this islet transplantation so that the individuals not only are insulin independent, but also they don't need to take immune suppressive agents every day that has its own long-term complication. The CAR T-cell technology is clearly, with our studies with Jim Riley and Mike Malone and Bohoj, it really is now putting us in forefront of inducing islet transplantation without immunosuppression.
Yeah. It's such a good point to see how an understanding in one area, say cancer, has direct applications to, say, diabetes or autoimmune disease of some other type. You know, I wanted to make sure our audience heard what you said, Ollie. You have figured out how to take islets and the cells that's comprised there. They make insulin from a healthy situation. You put them into a person suffering from type 1 diabetes, and they don't need to take insulin anymore. Now, that's remarkable. How close are we to that, having FDA approval for that, for our Penn technology?
Yes, we have completed the phase 3 trial. Everyone has heard about phase 3 trial, including for the COVID vaccine.
Yes.
And our results are the best in the world, frankly. We have been very open to help other centers to really tell what our secrets are. There's no secret. we have the best results. And I think the next stage is to go to the FDA. So this novel therapy would not be experimental anymore. It would be part of what we call standard of care. And a diabetic patient has an opportunity to undergo islet transplantation, essentially be cured of the disease. Yeah. And that involves enormous amount of support that I have been really wonderfully being given by the environment at Penn.
Well, yeah, you were talking about what's the secret sauce. Well, I know what the secret sauce is. It's you and your team and IDOM and the rest of the University of Pennsylvania School of Medicine, Perelman School, happening here, not other places. Ollie will come back. I'm sure there'll be questions for you on this incredible finding and advent of a real advance for type 1 diabetes. I'll introduce our next guest, who's Amit Bar-Or, recently, and not so recently, recruited to Penn. Hi, Amit, how are you? Good to see you. We knew each other from training many years ago, so it's great to see you on Penn's campus from time to time. Amit is the Melissa and Paul Anderson President's Distinguished Professor. He's a neurologist. He's an immunologist. He's working on another disease, not cancer, we want to talk about, multiple sclerosis. And he has a crazy idea that instead of T cells, there's another immune cell involved here. The B cell, the B lymphocyte, plays a significant role in multiple sclerosis. And that understanding can lead to new therapies. He's been working on it. Amit, tell us what's the news?
Well, thanks, Rob. This has been transformational in the MS field, both in terms of flipping on its head our understanding of MS, which, as you pointed out, we used to think of as a T-cell mediated disease, where the T cells are recognized as orchestrators of immune responses and they tell other cells what to do. And if not properly regulated, they may tell cells to contribute to injury of the central nervous system, such as what we see in MS. It's transformed our understanding of MS and a new wave of therapeutics that have been really gangbusters in the field. And this is another, I think, excellent example, as you've heard, of what you may learn by closely following individuals biologically when they're being exposed and treated to these with these experimental therapies. For me, the journey started some years back with a young woman, a 27-year-old, who had really bad MS. She was treated with every treatment at the time under the sun and kept on having bad attack after bad attack. We don't think of MS usually as a fatal illness, but she would have died from her MS. And what we ended up doing is bone marrow transplantation. We took her own immune stem cells. We treated her with very strong chemotherapy to get rid of her immune system, gave her back her own immune stem cells to make a new immune system. And remarkably, 15 years out, she's had not a single new attack and no MS progression. So to me, first of all, that was sort of a eureka of we can stop MS. The question was why and what was it about the BMT that we could elute that could be safer and more broadly applicable, because that's still a pretty dangerous intervention. And we measured in her and in others the difference in the immune system that comes back between the immune system that was there before. And it turned out that the bad T cell guys came back, yet there was no MS. What was different were the B cells. The B cells were back, they were different, and they gave us clues that they may be partly orchestrating other immune cell responses. And fast track forward, we've learned a great deal now about B cells and how they can control and regulate other responses, including of T cells and macrophage, and it's really the crosstalk between them that we now think of as we're trying to develop the next generation of treatments.
So B cells, maybe we didn't say it out loud, they're the cells that make the antibodies, but they have other functions too, which may be at the heart of MS. And we have therapies that get rid of B cells. We use it for B cell cancers. So can you tell us about how this idea became a therapy?
Yeah, so in fact, again, somewhat serendipity, somewhat being open-minded and looking at the biology. We borrowed one of the cancer treatments, the anti-Cd40 molecule, which targets B cells and B cell lymphoma and is very helpful there. But we used it initially in MS because people have known for years that antibodies are abnormally produced in the central nervous system of patients. Turned out that it worked really well in stopping new attacks. But when we measured the antibodies, they did not change. So B cells are important, but not because of the antibodies that they made. And that really was the foundation for a series of discoveries that we and others have had the privilege of contributing to that have taught us that in addition to being able to make antibodies, B cells do other things. They do other things in health, which are very important, and in different diseases. And it's our ability now to measure the responses of the B cells, the T cells, the myeloid cells at the same time in the same individual that, from a technology platform standpoint, is remarkably exciting to understand how people maintain their health, as John was saying before, and what goes wrong in a variety of different conditions.
Yeah, this is a great example of immune health, where we may be able to isolate our focus down to this particular cell, watch it in a patient who we're worried about for some reason, or intervene far before the damaging effects of a full-blown disease. I'm sure that we'll have more questions. There's so many exciting things going on in neuroimmunology. We'll hopefully come back with some questions. We just have one more panelist, actually. I told you it was a fireworks show. And I'd like to introduce our last guest, who's Caitlin Byrne. Caitlin is instructor in hematology oncology. She's a PhD scientist. She and I have actually worked together in the laboratory and continue to do so. Kate is a newly appointed fellow, a senior fellow of the Parker Institute for Cancer Immunotherapy, which some of you know about. Kate, you're studying pancreatic cancer. It's in the news. We're sad about Alex Trebek and so many others, Ruth Bader Ginsburg. This is a horrible disease. You've taken it on. You think the immune system, you think there might be an immune revolution for pancreatic cancer? Can you give us some latest insights?
Yeah, so as you mentioned, I work on pancreatic cancer, but I mostly actually work in the lab setting, so I use a mouse model. And my focus has really been understanding kind of the local tumor microenvironment, which is kind of an ecosystem that the tumor establishes for itself that has tumor cells, blood cells, and immune cells. And a lot of my work has been focused on understanding how the tumor directs the development of this ecosystem. And by directing that, it determines which immune cells are present and therefore if it's sensitive or resistant to immunotherapy. So our work in the lab has really focused on understanding Cd40 stimulation, which is a molecule expressed by dendritic cells and macrophages and B cells that we've talked about already in this program. This is all upstream of T cells. So by stimulating Cd40, we can actually drive a T cell response. And this has been working very well in the mouse model. And then one of the benefits of working at Penn is that we can bring those data to the clinicians. We're able to set up a clinical trial, and then we actually get the samples from the patients back into the lab. We can understand where the biology overlaps, and then we can modify our mouse model to better understand exactly what's happening in patients and further refine treatment options.
Yeah, it's all about making, when you do go to a clinical trial, that you have the most robust data to support its success, just like Drew did with the SARS vaccine, with the COVID-19 vaccine. We were able to leverage your work through the Parker Institute to conduct a clinical trial. The results are really, really promising. The paper has just been accepted for publication. We'll talk about that another time. But while I have you, I want to ask. So, you're the most junior rank of all the people we met. We met a lot of endowed, chaired professors and so forth. But you're the future, and your colleagues are the future. And the bridge to independence is a tough. It's tough. It can be tough. How is this grant that you got giving you a path forward?
Yeah, I think it is very tough. I think it is even harder, of course, with COVID and other stressors on the economy. But definitely, the Parker Institute really leverages funding and collaboration. And the fact that we were able to take data from the mouse model in 2016 and design and complete a clinical trial within 4 years is really amazing. And to have the data from the patients already analyzed from the phase 1b portion of the trial really allows us to move forward very quickly to the next iteration of this. So in 2021, we're hoping to open, the next level of the trial for more patients. And I think that that's only possible by really having the Parker Institute bridge the gaps between where we don't have fundings or we don't have access to reagents from companies, collaborations. I mean, this is a nationwide trial that we did, and this really is possible by having this type of access that's made possible through places like the Parker Institute. It's really amazing.
Yeah, it's a different kind of funding than like NIH, which is a little more traditional. This is more shoot for the stars, and you've done a great job with this. Well, so that's our panel, and I want to circle back and go to the audience's questions. Many have poured in, and we're going to see if we can do this like a radio talk show, where the person to answer the question will show up on your screen. There's some of them, we will see. So, let's go back to COVID-19. One of the questions for you, Drew, see there's Drew, is about this. It has to be kept super cold. We hear a lot about this. And so can you tell us what's that all about? Something about RNA needs to be super cold? And what can we do about it?
So it's actually not the RNA. It's the lipid nanoparticle, the lipid carrier. And that's really a developmental thing. So once the vaccine works, now we have to figure out how to keep it stable at refrigerated temperatures, which we're out to about 2 months stability. We've also figured out how to freeze dry the vaccine, so that I think that's a minor inconvenience for a vaccine that was made in 9 months.
I'm glad you mentioned the lipid particles, because that was another question. Let's just clarify the whole formulation. If you had a microscope, what would this look like? RNA with a lipid molecule, a lipid covering?
Yeah, so it's actually a solid nanoparticle that's about the size of a virus, where the RNA is on the inside and the lipids form a shell around it.
Excellent. Yeah, as far as I know, do we have any drugs that have that kind of formulation, or is this going to be brand new?
So actually, there's an FDA-approved drug that uses lipid nanoparticles to deliver siRNA for a form of genetic amyloidosis.
Yeah, that's so that's encouraging that we've in some ways been down this road before. Another question, Dr. Naji, Ali Naji. We have a question. Could you describe, like, what does it look like to get an islet cell transplantation? Like, you walk in the hospital room and what goes on? Is it an operation?
Yes, it's done through a very minimal procedure without requirement for anesthesia. Actually, routinely do it in the Department of Radiology through a small needle that puncture, and we inject the islets into the liver. However, again, at Penn, we have now reported just a couple of weeks ago, we are able to implant the islets under the skin with excellent survival. So this is a very new frontier in terms of being able to biopsy them, looking at the biology of it, and that has excited a lot of investigators to be able to look at the biology of it in vivo, in a human being.
Yeah, that's continued innovation. We have a question from earlier, phoned in through the portal, maybe for Sar and Shannon. Immunotherapies, CAR T cells certainly have a pretty high price tag. So cost and affordability, it's an issue. How are we going to deal with that? I'll let each of you have a comment.
Shannon, you go first.
Okay.
Yeah.
So it is an issue. Absolutely agree. And I think that there's two aspects to it. One, I think if we are able to start moving these therapies earlier in therapy, we can sort of put this in perspective that the cost of this is likely very similar to the additive costs of many of the years of chemotherapy that we give, or the additive costs of bone marrow transplant and potential toxicity down the road. So I think in perspective, it seems like it's a one-time price tag. It seems like a lot, but it is fairly similar. But we still need reduce that. And I think there are lots of technologies that will aid us in being able to advance the manufacture of these cells so that it is not nearly as costly for every single run of CAR T cells. And there are lots of technologies looking at, can we potentially make CAR T cells from healthy donors and have a large supply that can be given to multiple patients.
Off the shelf type of approach. Sar, do you have anything you want to add to that? Otherwise, I have a question for you.
No, I completely agree with Shannon.
Yes, good for you. The question is about commercialization. You mentioned it, and it sounded key and fundamental. Of course, we know we partnered with Novartis to bring CAR T cells. So what's that road look like, and what are the pros and cons?
So to get as expensive as we have heard CAR T cells to be for patients that we want to prescribe them to, the path to getting FDA approval for something that starts off as a glint in a scientist's eye to an approved drug is the price tag is enormous, and it's impossible to do that with research grants alone. Or almost impossible. And so very often the path to commercialization does go through collaborations with industry, collaborations with biopharmaceutical companies in order to draw those resources that are needed to test the concept in humans.
Yeah. And of course, I think Penn and Penn Medicine recognizes that, and it has a number of structures. You've utilized them to bring those types of resources, and we see what's happening. Kate, there's a question about pancreatic cancer vaccines. So we talked about a vaccine for coronavirus, there's a question about cancer vaccines, and in particular in pancreatic cancer. Are there some options for a cancer vaccine?
For pancreatic cancer, one of the problems with it is that unlike melanoma or other very highly mutated cancers, pancreatic cancer is kind of middle of the road or even lower. So that just presents a little bit of a barrier of knowing what to target for a vaccine. We don't have necessarily great targets. But the other main problem is that we need to get those T cells into the tumor. So it's very similar to what we've talked about for CAR T cells. There's still a problem of getting the T cells that you generate from the vaccine into the tumor site itself. So I think there's potential to explore vaccines, but they kind of need to still be in combination with other treatments that can really remodel the local tumor ecosystem to allow T cells to want to get in there.
Yeah, the immune system has to see something foreign for the craziest response. Coronavirus is foreign, so we see a great immune response. Pancreatic cancer, unlike some other tumor types, doesn't have quite as many mutations and doesn't come across to the immune system quite as foreign. Plus, it has this shroud around it. But it does raise a question whether or not this RNA technology we've been talking about today, Drew, is there a cancer application, do you think?
So there definitely is. We're working on some cancer vaccines. BioNTech, the company that made the coronavirus vaccine with Pfizer, has a clinical trial with a personalized mRNA vaccine that's ongoing right now. So and they've had wonderful results in their preliminary studies.
In many ways, if this vaccine we talked about today makes its way forward for coronavirus, it will be a whole new level of energy put forth for RNA vaccines in cancer, I would hope. So a question that was put in earlier last week, maybe I'll ask Joe about this, because Joe, you're newly hired, newly minted independent. The role of the institution. We've talked a lot about Penn, and this is homecoming. What makes Penn so special? How is Penn supporting you and your colleagues in this really complex stuff? Can you speak to that?
Yes, absolutely. So with Penn, I think it's one of the very few places in the world where we can go from proof of concept to a pilot study, a pilot clinical study, or a phase 1 clinical trial in a year. So there are, very few places that can do that. And the Abramson Cancer Center, the Center for Cellular Immunotherapies at Penn, supports these pilot studies. They encourage this rapid translation from the bench to the bedside, and sometimes back to the bench, as we've heard.
Yeah. One of the most common phone calls I get from other people at other institutions is they'll say, "We want to get into the immune therapy, cell therapy space. Can you tell us the secret?" And I said, "Well, we started 20 years ago with ongoing sustained vision from the institution, and that's why we're all here." And I have one more question. This will be for Amit, and then we're going to go to a lightning round and close it out. But the question is, people were listening very carefully to your answer about multiple sclerosis and autoimmune disease. What about other autoimmune diseases? Can you move into IBD, inflammatory bowel disease, or into Crohn's in particular, can we leverage what you're learning in MS for other diseases like that? What do you think?
I think the answer is a resounding yes. It's actually one of the main reasons that I ended up coming to Penn for what you had heard from other colleagues in terms of the collegiality and the collaboration. There's very much an opportunity in what can my disease, so to speak, teach you about yours, and vice versa in the context of immunology. And indeed, we have active studies and collaborations across other neuroinflammatory conditions, like with Alice Chen-Plotkin in Parkinson's disease, but also in psychiatry with Raquel Gore, maternal inflammation during pregnancy and the impact on the fetal brain development with Michal Elowitz, with Ali Naji looking at MS, directly comparing it to type 1 diabetes with another Penn special, which is looking at a hidden immune compartment, the thoracic duct. And these cross-disease comparisons and establishing the platforms where we can apply the same measurements to people with multiple different conditions as well as healthy individuals is really at the core of what we're talking about in terms of a new immune health initiative.
Well, that's awesome. We have just a few minutes left. We're going to shift to the lightning round. Lightning round means lightning answers. And so I'm going to ask you all the same question and just go around. I'll start, since Amit, you were speaking, I'll start with you. What do you need to take your work to the next level? What do you need?
We need a technology platform that will allow us to make these measurements, all these different cell responses at the same time in a tiny amount of blood, let's say. And an individual who would be what we call a systems biologist, who knows how to deal with the huge data sets that emerge and how to actually interrogate them in a meaningful way and make them available to other people across disciplines to use as well.
Kate, how about you? What do you need?
We always need a little bit of pilot funding to get just one piece of data to know kind of, should we go or no go on an experiment, or one piece of equipment to kind of increase our throughput. So just little extra help sometimes.
A little extra help. Joe, how about you? What do you need?
Human capital is essential, so we need to do our best to recruit new talent on a consistent basis and retain the talent we have, especially during this pandemic.
That word I know from the job I do, retain. Everyone's after our talent. They're all after you guys. Don't answer any of their emails. Sar, what do you need?
We need space. So we can have all the human talent and all the funding to do research and the ability to do clinical trials, but we also need, and this seems maybe a little bit humdrum, but we actually need more space to park those people that do the research. It is a big ask, of course, but more lab space, a building to put the lab space in, that's what we need.
Shannon?
Infrastructure and trial funding to allow for a really nimble clinical trial regulatory framework so that we can advance these novel therapies and get them to patients quickly.
It's so expensive. You're absolutely right. Ollie, what do you need?
I thank you. I need two things. One is I really would love the Penn family and alumni to join us in a very special Penn Diabetes that we have arranged in January 27 and 8th, and also join us, our Leadership Council. but it's taken us 20 years to perfect how to isolate islets from pancreas. It's an art and science. We have now enormous amount of talent and expertise, but I need a facility, which is called GMP facility, good manufacturing practice, to isolate this unique cell on Penn campus so that we can really now enroll larger participants and patients.
Yeah,
Thank you.
Cell manufacturing, absolutely, it'd be a game changer. And finally, Drew, you started us off with incredible news and such great promise. But from your perspective, what do you need?
So I think all researchers need sustained funding for basic science research.
I couldn't agree with you more. That's a great lightning answer. Basic science, everything we heard about today came from a basic science discovery. No one's just making this up. Kate is discovering, Shannon's working with folks in the lab, Sar has, just on and on. Until you discover, you can't translate. It's a great message to end on. I would like to thank everyone who joined. It's nearly 200 people from all over the country, all over the world. Thank you for joining Penn Medicine Homecoming and the ImmunoRevolution. Ideas, the best is yet to come. We're going to tackle coronavirus. We're going to tackle cancer and MS, a place where it's going to happen, the Penn way. We're going to solve these problems, and we're on our way. With that, I'm so happy you joined us here back at Penn. You're home, and we'll see you again another time. Thanks very much.